Hey everyone, welcome back. We're going to be talking about photosynthesis in this last video. So now we're talking about producing organic molecules from inorganic materials like carbon dioxide and the utilization of sunlight energy. So sunlight energy is trapped via different wavelengths of sunlight energy are trapped via pigments. Pigments are found in structures we call photosystems. Photosystems are then found within membranes inside of either plants, algae, cyanobacteria, or anoxygenic bacteria. The different pigments are going to differ depending on the organism. So in our aerobic organisms, we use chlorophyll right here. In anaerobic organisms, they often use a substance similar but not the same, bacterial chlorophylls, which pick up different wavelengths of light. And then we have accessory pigments like carotenoids or phycobilins that are utilized at different times of the year. So we have two major reactions in photosynthesis. We have the light-dependent reactions, and then we have the light-independent reactions. So light-dependent reactions occur within our cyanobacteria and photosynthetic eukaryotes. And there are two different types of light-dependent reactions. So we have a cyclic photophosphorylation and non-cyclic photophosphorylation. Both of them produce ATP. You see this. Both produce ATP. But non-cyclic also produces NADPH. Okay. I'm going to go to the next slide to show you these because that's where I have the pretty picture. So here we're looking at cyclic and non-cyclic photophosphorylation. So when we're looking at non-cyclic photophosphorylation, we start in photosystem 2. I always like to explain that there are sometimes naming materials can be confusing. So we talk about photosystem 2 first and then photosystem 1. They were named in how they were identified. So photosystem 1 was found first and then they found photosystem 2. That's the only reason they're named this way. Photosystem 2 is the first photosystem in the non-cyclic pathway though. And so in this reaction, we're going to take water and we're going to split it, producing hydrogen ions and releasing electrons to move into the photosystem. Oxygen is a waste product of photosynthesis and it is released into the environment. So here we have electrons, and I'll just put a little electron here, moves into photosystem 2. Sunlight energy comes in and it traps the energy. This energy excites the electrons. The electrons, as they get excited, they move up in energy levels and eventually they're going to jump. When they jump, they move into an electron transport chain. So this electron transport chain shuffles the electrons from one carrier to another and pumps these hydrogens that were released from the splitting of water across a membrane. So that produces a proton gradient. The proton gradient then, since we have more hydrogens on one side of the membrane and less on the other, they want to move back until they're at equilibrium. They flow through an enzyme called ATP synthase. Very similar to aerobic respiration. Okay. And so we produce ATP in this process. The electrons, as they move down in energy, they move down and down and down. They eventually end up in reaction center of photosystem 1. So I'm going to quickly erase this so you can see this side. All right. So they move into this photosystem 1, and again, sunlight energy comes in, activating the electrons. The electrons rise in energy, and they jump again. And in this case, they jump to an electron carrier, NADP. So if you remember, this is NADP. NADP reductase actually takes the hydrogen and attaches it to NADP, producing NADPH. And this NADPH is an electron carrier in a biosynthesis pathway. So remember that. Biosynthesis, we're building something up. Photosynthesis, we're building up organic molecules. If this is the cyclic pathway alone, so we have both, I'm going to erase all this. Then we're looking at electrons that have moved into photosystem 1. When they jump, when they get excited, they jump, they move into the electron transport chain, which produces ATP, and then they move down in energy until they can move right back into the reaction center of photosystem 1. And sunlight energy comes in, they get excited, they jump, and we just keep going through this cycle here, and we produce a lot of ATP. So non-cyclic produces ATP and NADPH. Cyclic produces ATP alone. When we talk about the light-dependent reactions with anoxygenic organisms, then we can't use water as our donator for electrons because oxygen would be the byproduct. If you're anaerobic, you don't want to have oxygen in the environment. Typically, anaerobic organisms don't do well in the presence of oxygen. So they're going to use other materials like hydrogen sulfide or nitrite to get rid of or to donate hydrogens. They also tend to produce less ATP, and because of that, they're not going to gain as much carbohydrate for the same amount of work. Okay, hold on. There we go. So the next step is the light independent reactions, which also are thought of as carbon fixation. We're going to take carbon dioxide and we fix it to a 5-carbon molecule, producing a 6-carbon intermediate that is unstable. So in this process, we have to utilize the ATP that was produced in the light-dependent reactions, and we're going to utilize the NADPH that was produced in the light-dependent reactions. So in general, the Calvin cycle is how we produce this, and we're going to first take carbon dioxide and fix it. We then reduce it, and thirdly, we regenerate the original compound. So this is a cycle, which means you start and end with the same materials. So here is basic Calvin cycle. Here we have 3 carbon dioxides being released. So each of them is donating 1 carbon. Each of them will bind to a 5-carbon molecule, and this 5-carbon molecule is known as RUBP. RUBP bound to this 5-carbon molecule produces a 6-carbon intermediate that is very unstable. It immediately breaks down into 2 3-carbon molecules called phosphoglycerate. These are then going to be reduced to glyceraldehyde 3-phosphate. And when you have 2-glyceraldehyde 3-phosphate, you can produce a glucose. So in this case, we have because we had 3 carbon dioxides enter. We end up with 3 6-carbon molecules, which split into 6 3-carbon molecules, which are converted to 6 3-carbon glyceraldehyde 3-phosphates. Only one of the glyceraldehyde 3-phosphates is actually going to be used by the cell. The rest, the other 5, are going to go into producing more RUBP, so that we always reproduce the RUBP. And so this is your table that shows the same thing. This is a little more simplified. It takes a total of 6 carbon dioxides to produce 1 glucose molecule, because glucose is a 6-carbon molecule, and each carbon is brought in, and we're binding it to this 5-carbon molecule here called ribulose 1,5-biphosphate, or RUBP. And so we're going to produce a total of 6 carbon molecules that are unstable, and that's going to break into the 12 3-carbon phosphoglycerates. We're going to then reduce the phosphoglycerates and produce 12 glyceraldehyde 3-phosphate molecules. 2 of these will get to go into producing 1 glucose, or which can then be used for whatever we want to produce. The rest go into reproducing the RUBP. All right, that's the end of this video. So I am going to stop this, and I will upload it, and I'll get all of these uploaded to your website just shortly. Have a great day. Bye.